Hello, my beautiful biology humans. Welcome to topic 2.7. This is a doozy. It's a long one. Definitely going to be two parts. First one has a couple experiments in it. So this one, part one's going to have a couple experiments that I would rather have you focus on the drawing than the actual words. So make sure you are listening when I go through the slides, because there's some stuff that I don't need you to write down, but more so understand the experiment. And then we're just going to do a lot of practice about this in class, especially with the proteins. So we have learned about DNA in our last lecture and how it's built and the structures and the parts. We have learned about proteins. Now we are going to combine the two, right? How do we get from those structures of DNA to the very complex proteins that we have learned about, and enzymes and amino acids and peptide bonds and all those fun things. So this is, we're going to be looking at transcription and translation. Again, your experience in 9th grade was so varied that I don't know where you and when you learned about this. So we'll just be going through this step by step, talking about the enzymes that are involved, talking about the steps, and really focusing on the vocab for this. Okay, so we're going to do up to transcription for part one, and then the rest of it will be in part two. Okay, in the first part, we're going to go through what is called one of the only three. There's three models in science that are that really kind of what I'm saying encapsulate everything. One is the natural selection model, the one is the cell theory that you've already been through, and this is the third one. This is the central dogma, right? And so the central dogma, it really is just saying this down here. How does DNA get to RNA, get to proteins, like the process in which that happens, right? So the flow of genetic information into something being built. Okay, now the three components of the central dogma that you need to know: DNA is copied in order for new cells to be made. We've already kind of talked about that in the cell cycle, right? So one of those checkpoints is, do I have my DNA replicated? Yay. The second part there is the process called the transcription, which is an mRNA, right? It's a part of when we looked at nucleic acids, right? The messenger RNA. And I briefly explained there that scribe means to write, right? So the DNA may code for those RNA transcripts via this process. And remember that the RNA, if we're going all the way back, that messenger just takes a little part. It just opens up that gene that it needs to copy. And then the last component is that mRNA codes for the translation into the protein, right? So the mRNA, it goes along with that little amino acid. We have the peptide bonds that form those, the bonds between the amino acids, and that ends up creating our protein, right? So DNA to RNA to protein, those process of replication, transcription, translation, right? That is the central dogma that we'll have learned about a little bit and we'll be learning about in this lecture, okay? Comparison of cell processes. So let's look at these three things in a when it occurs, where it occurs, and my key enzymes and the point of each, or the function of each. And let's start here. Okay, so replication we've already learned about. Let's just review that S phase, right? So when it's getting ready to divide, that S phase is when the DNA actually replicates. It is for DNA replication that occurs in the nucleus, right? So we've learned about that organelle and where that occurs. My key components are enzymes. We're going to learn about those enzymes today, but you should know that, like, I love this. In order to make proteins, you need an enzyme protein to be part of that process, which is very confusing, right? And the function is to just duplicate genetic information, exact copy, before the cell divides, right? It needs to divide into two. Okay, transcription that we'll be talking about today is happening all the time, right? They're constantly writing things down, transcribing, and then continuously translating. So this process happens all the time, depending on what our body needs, what's happening with breaking it down, right? Depending on what's going on in our body. Transcription still happens in the nucleus. Remember, we talked about how those nuclear pores are really important. We still have enzymes being the really key component of that process. And its function is to produce that RNA that's going to transcribe that DNA sequence, just a little part of it, okay? Translation, again, happens all the time, but this is happening in ribosomes out in the cytoplasm, right? So it's either at the rough ER or ribosomes floating. And so this process, right, that little code goes out of the nucleus and goes to the cytoplasm, to the ribosomes, to make the protein, right? So we do need to know where these things occur. We need to know the key components and then when, right? DNA replication, we've already covered that, but these two are going to be our new ones, and we need to know their functions and where that occurs. All right, so let's talk about replication. Yay, making an exact copy. All right, big concept here, big keyword, semi-conservative, right? Semi-conservative, to stay the same and then half, right? Semi-annual sale, ooh, right? Half a year. So semi meaning half, conservative meaning staying the same, okay? So when DNA is replicated, it is unwound first, and then the strand is copied from the middle out, right? So it opens here, right? So you can see my DNA's template. So it opens here. I have one original strand on each one and then one new strand, right? So I make an exact copy of it, but by having half in original and half new. So this process is called semi-conservative. Half is staying the same, half is new, right? This occurs because of my base pairing rule, my ATGC. So if I split something apart, but I still have that base pairing rule, I'm going to get the same result, right? So my old strand and my new strand are going to be identical. However, they only have half of that original strand, right? So semi-conservative, half old, half new, splitting apart. We'll talk about those enzymes here in a second. Okay. The experiment, do not write this down. Stop. Do not write this down. Okay. What I'm going to have you guys do is on the next slide, there's all these visuals, right? So this is actually put into, I'd rather have you have the visual of this experiment than this written down. But here's basically what happened. Like they trying to figure out like how this works, a semi-conservative. The Meselson-Stahl Experiment, was looking to prove that semi-conservative, right? Half old and half new. So what they did is they marked nitrogen bases, right? So they're able to put a radioactive isotope incorporated with the DNA, and then it was induced to replicate, and then was spun around, and then was looking at the results. So after that first division, right, we have both of those present, but after the second division, only one of them was, showing that like it kept splitting apart. All right, so I'm going to show you the next one. I'm going to have you copy this down, right? And then we can go back and add some words to kind of add, add some, what I'm going to say, words, explanation, that was the thing, to the pictures, okay? All right, so here are my actual observations. So if it would have been conservative, so if I have, this is after the first division. So after the first division, we had both of these markers present. If it would have been conservative, right, I would have had these two. It supports the semi-conservative, meaning like it makes an exact copy, right? And the dispersive is when they completely separate and go apart. So after the second division, right, my actual observations, I got N14 only. This is the centrifuge where it goes into two parts. And then this supports that conservative because I only have that one splitting apart. We'll do this in class with splitting DNA and showing you guys in the centrifuge. And sorry, this is that disproves the conservative and shows the semi-conservative, and though disprove the dispersive. So when we're looking to the semi-conservative model and this experiment, right, the Meselson-Stahl experiment, we'll look at like that experiment actually acting it out. I'll have you guys do those replications so you can actually see how and why that would work. But the second division, right, looking at second, is what disproved. So the first one disproved the conservative, but the second one disproved the dispersive. Okay. All right. So here's how it actually replicates, right? And so here's where we get our first like important enzyme that we need to know. So my intention is to replicate, make an exact copy, right? Remember that right before they copy, they condense into chromosomes, right? We looked at that before cell division. They condense, right? They go from those long strings to being spun around those proteins. And then this is this first enzyme we need to know about is enzyme is helicase, DNA helicase, right? So this helicase is what breaks those bonds down the middle. Remember that the hydrogen bonds keep those nitrogen bases apart. It's the one that breaks them up, okay? The next enzyme that comes is DNA polymerase, and this is the one that attaches the new hydrogen bases, right? So helicase breaks them apart, polymerase puts them back together, right? I always like to say helicase raises hel, and then poly, good old Polly, comes around and she puts the strand back together. Okay, here's a short visual of that. I'll just show you those 15 seconds. Clear the screen back. There you go. There's some fun music here too. Ready? There you go. Helicase. Look, I'll like narrate it for you. breaks the strand apart. Those the new bases are added, and then polymerase comes and bonds them all together, right? New one, woohoo, right? So I get that one, breaks it apart, and then it comes back here and puts those bonds together here on the outside so that those nitrogen bases are all connected, right? Those phosphate bonds. So helicase breaks apart, polymerase puts it back together, okay? This unwinding, so a little bit more about helicase, right? Oh, I just said this, but you can write it down now. So how it unwinds is by breaking those hydrogen bonds that exist between those pairs, right? So what it's actually pulling apart are those hydrogen bonds. And then I get these exposed, right? This inner part exposed so that new bases can fit in there. So it takes that tightly wound, it unwinds it, right? And then it separates it by breaking those hydrogen bonds, okay? Then polymerase comes in, right? And then this is the enzyme that comes apart, that comes in and it adds the nucleotides, right? So the nucleotides like line up because they pair together. Remember we looked at like the different rings. Those exposed complementary bases are available, so they come up, and then the polymerase is what makes the covalent bond on the outside, right? So we talked about that covalent bond that occurs between the nucleotides to form the new strands, right? A much stronger bond than the hydrogen bonds. Okay, DNA polymerase 3 is the thing that synthesizes the new strand from the template, and it uses covalent bonds. We will talk about the 5' to 3', right? 5' to 3', that's the direction that it moves, is 5 to 3. If you go all the way back a couple lectures ago, we looked at how those rings have the carbons that are numbered, right? 1, 2, 3, 4, 5. Just know that the bases are added on a 5 to 3 direction. Right? So it's actually opposite directions because they are moving, those are lined up in opposite ways. Okay. All right. Polymerizations. Oh, right. That process, right, is what's called, like, that whole process is called polymerization. And that's when the nucleotides align to the complementary, they're joined a growing chain, and then it cleaves the two phosphates and releases the energy. And that energy is used by the enzyme to create a covalent bond with the nucleotide chain. Wow, that was a lot of fun words, right? So new nucleotides, the new ones, align with their complementary bases, right? And they partner as the deoxide to the triphosphates, right? So that they're lined up. And then when they're joined, right, to a growing chain, the polymerase cleaves those phosphates, releases energy, and that little energy comes down and makes the covalent bond with the phosphate group, the phosphodiester bond with the nucleotide chain, right? I feel like that one needs a visual. We'll look at that in class, all right? Polymerization. Whew. Here is the process by which, like, we can artificially, so the polymerase change reaction is an artificial method to show, like, or process to rapidly amplify DNA sequences. So we can actually, they can actually increase this happening, right, to amplify or move up or increase this replication, right? So polymerase chain reaction is made short by PCR, and it has these three different steps, right? So you can see this one here: denature, annealing, and elongation. All right. So this process of PCR, before I go into those, 30 cycles generates more than 1 billion copies, right? Here's how it works, right? The denature step, it heat separates the strands. So there's no helicase involved, but there's a certain temperature found where the strands break apart, so 95 degrees. The annealing is when the primers are designated the copying sequence, right? So they look here, they're priming to copy the sequence and add the bases. And then the elongation process is when the copies the sequence. So that's the one that actually goes around. Look at the different temperatures too, the 95, 55, 72. So very specific situations in which these occur, right? So, and they're kind of mocking, or what I'm going to say, copying what the enzymes do in an artificial model that creates a lot of DNA or right away, right? So copying the DNA. So denature, breaking apart, starting to add the bonds here, and then putting them back together with that elongation. Okay, so the use of polymerase produces multiple copies by rapidly moving along the polymerase reaction. Okay, All right, part 2 will be our transcription, so how we actually go from having that DNA to making proteins. But for this part 1, make sure we know all those key terms. Make sure you guys know semi-conservative. Make sure you have a good idea of how the enzymes that are breaking apart and how this artificial process of PCR has changed the rate at which we can replicate DNA. All right, people, I will talk to you later. Have a fabulous day.